For developmental biology, in general, has been quite essential for development, for biology. Developmental biology is one essential aspect, as was shown in the Nobel Prize to Christiane Nusslein-Vollert and Wieschaus and Lewis. So, I mean, they, for example, set up the idea of how is the body patterned during development. So, if you think of that we are segmented in our structure, so we've got our vertebrae, for example, which reflect that very nicely in our segmental nerves, then asking how this pattern is set up was started in Drosophila. So people basically took the segmented pattern of the fly, of the cuticle actually, of the outer shell of the maggot, and then they asked, can we make mutations in which these patterns are affected? And they found a whole range of those, started studying them very intensely, and when then people like Peter Groos, for example, took that over and looked into mice, they suddenly found the same genes. They found that the same genes are doing very similar jobs. So I think as long in biology, as long as you formulate your question as a principal mechanism, or looking into principal aspects, principal mechanisms of biology, is a very efficient and very important model organism. And of course what makes it so important is the fact that we know from the sequenced genomes, which have both been published, of course, of humans and flies, that an enormous amount of genes has very high homologs in the human genome. And as such, if one takes the statistics, I think it's about 70% or so of genes have conserved homologs. And it's a very high percentage of genes that have been linked to diseases. For example, be it developmental diseases, be it any kind of other diseases, which have been considered to be homologous enough to use them for translational research. So explore the principle of these genes and their function in the fly, and then look in the human or in mammalian models whether we can reproduce these results, whether they're true also in biomedical contexts. In my current research, it's key. So I'm a strong believer in that complexity is can much better be addressed in flies. So we work on brain development and regeneration, neurodegeneration, and they're looking into the cytoskeleton. So that material inside the cell that basically drives its dynamics, cell dynamics, like microtubules and actin. Now, all the regulators, or the majority of regulators, highly conserved between flies and mammals or humans. But the copy number of genes is much lower in the fly. So if we now if you think of neurodegeneration, for example, we know that molecules like tau play an important role in many forms of neurodegeneration. Yet, if you look in the literature, tau is up to now very little understood in its function. Although many years of research have gone into that. Now, Drosophila has a fairly well conserved tau gene. And in our research, for example, we are uncovering at the moment very interesting overlapping functions between other classes of genes with tau, which have never been considered in those contexts. So there is certainly possibilities that while doing this kind of research, you stumble or you discover new areas and new genes which have to be linked into the consideration of that kind of research, and therefore can enormously drive the advance of that research. Let's say the ethical aspect makes it very easy for us, so we don't have to go through all these long training aspects like people working with mice or with other vertebrates. We don't have to write long license forms to get our license and permissions, because flies are officially not considered being animals legally, but of course, some of these aspects, especially in modern science, it tends to be no longer enough to just work on Drosophila. But it makes a case stronger if you can really demonstrate the translational aspect into mammalian biology. So if you can demonstrate that what you find in Drosophila really is relevant in the higher organisms, that would make your output more valuable in modern science. I wouldn't say that it's more valuable necessarily in the biological sense, but in the, let's say, social evaluation of your work, it would be much more important. Insofar, it is very difficult to just restrict to Drosophila. We have done ourselves some work in mice to do some translational studies, and it is constantly in our mind that if we have certain findings, that we talk to colleagues in the fields who are working on related aspects and ask whether they would be interested in collaborating and testing some of the mechanisms that we find, which is usually the best way forward, because maintaining all that expertise alone in the lab is very difficult. So running various model organisms in parallel is possible for very large groups, but in the modern funding landscape this has become very difficult. Yeah, of course. I mean, we would be lying if not. So, as I said, it is always about the principle. So as long as we talk about biological principles, and I've showed you some films before, for example, of behavior, where you see aggression. Aggression is an inherent need, obviously, biological need for survival, and you see that these things have been conserved even in the fly, right?
Yeah.
So it's not that different seeing fighting flies on an arena under the camera, under the microscope, compared to fighting bulls in Africa. So it looks very similar. So if you think about that, then of course you would demand that there are certain traits which have been conserved. Now these are very complex questions, but if you think of other behavioral aspects, like for example circadian rhythm, so the fact that we get jet lag is because we have an intrinsic rhythm of about 24 hours. So if we would lock you into a dark room and you could switch on the light whenever you wanted, you would actually follow a 24-hour rhythm. And the flies do the same. And it's in flies that the first genes have been discovered that regulate this behavior. And actually it's up to now that the fly is the leading model organism for understanding the principles. And it has been shown that the principles are certainly the same. Now if you come to the limitations, of course our brain is much larger, and there is much more aspects to it and much more complex behavioral traits that have to be analyzed. If you now look at, for example, the suprachiasmatic nucleus, which is very important in the circadian rhythm of humans or of mammals, then such structures don't exist in the fly. And there you have huge networks of cells interacting rhythmically with each other, and there are hormonal systems underlying this. These systems, of course, are then not available in the fly. But as long as you ask for the key mechanism of what drives the clock, the fly is fantastic, right? And it's actually one of the leading model organisms that we have. In general, I guess we would be far behind, very far behind. The key, I just told you, we have 4 Nobel Prizes. One of them is in general on the chromosome and gene structure from Thomas Hunt Morgan, and there's one on mutations. I mean X-ray and the role of X-ray mutations. I think that was Muller. Then we have the one on patterning, so the development, which revolutionized the field in developmental biology, and this year's Nobel Prize is on the immune system, so showing principal proteins which are key to the immune system in the fly, which have then been shown to be essential in humans. So I guess that many of these discoveries would have come much later, and many of them would not have come, because what the fly does is, through its simplicity, through its very easy way to deal with it, through the speed in which you can do analysis, through the low redundancy, genetic redundancy in the system. So in mammals, for example, you very often have 3 similar genes, in Drosophila you have just 1. So in the mouse you have to take 3 genes out in parallel, in Drosophila just 1. So that makes things much, much easier. And so basically having these biological and mechanistic principles worked out in a simple model organism gives you now an intellectual framework into which you can embed all kind of work that you would do in mammals or studying disease of humans, right? So even if the concept may be different, it is still a facilitator of driving, intellectually driving that process. So I would say that we would be far behind, I guess decades behind in our research. The Drosophila has many features, and this unfortunately, when people work on it, turns out to be the case in many aspects. Well, it has very strange features in early development. It has very odd features in the cytoskeleton, for example. Is one of it doesn't have intermediate filaments, which is in many other invertebrates and in all vertebrates. So this can be challenging. This can be interesting, because you can ask about how important are these features really, or how much are they simple facilitators of processes that will function anyway. And you could ask of how can an organism deal with the absence of such mechanisms. So it can be intellectually very, very interesting, but certainly if you want to use it as a model, there can be drawbacks. And other insects, for example, may have been better. It's hard to say, because research over this long time period, in which of course once you learn something you can use the learned in order to build on it in your further research. So you've got a positive feedback loop that very much enhances advance. And this very much enhanced advance, of course, in Drosophila is unique, and there is hardly any other model organism or invertebrate model organism apart from C. elegans which would have this effect as strong as in Drosophila. So it is not replaceable at the moment, and it will not be for a long time, I would guess. It may have been beneficial to have started with other insects which don't have some of these specific aspects, but which then may have other disadvantages, like for example longer generation time, or perhaps not be as manipulatable. So I guess overall the choice was good, was very helpful, and I think it will be unreplaceable for a long time.
Great. I think that's everything I want to ask.
Yeah, good.
Thank you very much.
Thank you for listening to you.